right. Well, welcome everybody. Thank you for coming tonight. I'm Brian DeSantis. I work for UC Cooperative Extension Office in Reading. I'm the Forestry Advisor for Trinity County. If you haven't gotten these handouts, there are 3 maps that pertain to Southern Oak Death in Trinity County. There's a handout on the quarantine and some sample handling instructions. First, we're going to have Matteo Garbolato, who's a specialist at UC Berkeley, with more sudden oak death information, and then I'll follow him by just kind of briefly discussing some of the sudden oak death maps and handouts that are over there. So I'll hand it over to Matteo.
Okay, so please, there should be a sign-up sheet going around.
Oh yeah.
Wherever that sign-up sheet is, do put all of you, even if you came as a couple, put both your names. You can put just one email. The email, we sell the email to make money. I sell it privately to the highest bidder. So you'll be flooded by thousands of emails. So about 2 hours after the meeting. Now, we only use the email to let you know when the results are in of the survey and when the next meetings are going to be, nothing else. If you don't have that email, then we have no way of communicating with you. So. Again, I'm Matteo. Everybody knows me by Matteo, but my last name is Garbolato. I'm UC Extension as well, but I'm based on the campus, and most of the forestry specialists within the UC system are based at UC Berkeley, so that's where I am, and I'm also a professor there. And I hope that you're not going to be disappointed today. I don't think so, by looking at your eager, expressions, because this is not your traditional educational seminar where I'm just going to talk, blah, blah, blah. This is actually about doing something. And what we're trying to do here is we're trying to engage you and basically use you as free labor to determine the distribution of the disease in your county. And the disease we're talking about is sudden oak death. It's a very important disease. It's highly regulated in this country and in other countries. It affects the economy of the areas where the disease arrives because a lot of things that normally you could do, you can't do when you have the disease. But today we're not going to talk about regulations, and we're not going to talk about the biology so much of the disease. We're really going to talk about how do you go about surveying an area, and how do you collect samples so that they can be processed in a good way and they can bring results and increase our understanding of how far the disease has gone in Trinity County. And the activity is called Blitzes, SOD Blitzes. This is now the 8th or the 9th year that we're having these. And they're not called Blitzes because of any military association, so don't worry about it. We're not enrolling you in any branch of the Army, Navy, or Air Force. They're called Blitzes because we like people to survey an area in a relatively short period of time. And that's relatively important for 2 reasons. One, because of timely collection and processing of the samples ensure the best results. And the second one, because if everybody collects samples at the same time, there's a lot more that we can actually understand about how the disease is operating in any given area. And this is organized by my laboratory at Berkeley, but it's also very heavily supported by the California Native Plant Society. And it's considered now one of the largest citizen science efforts in the country. It's also one of the longest one that has been going on in forestry. And I put down here some URLs for websites. I will show this again at the end of the presentation. But some of the information and other information that you may be interested in is present in these URLs. And again, I'll show it at the end of the talk. So very, very briefly, a little bit about the organism and the disease that it causes. The reason why we're all here is because sudden oak death is caused by an organism that is exotic. So it's not a native organism in California. We think it may have come, we know it came probably in the late 70s and 80s into the ornamental plant industry throughout the world, both in North America and in Europe. And we think it escaped in the late 80s from nursery settings and from private properties where ornamental plants have been planted. And from there, it started to spread in our native ecosystems, especially on the coastal ecosystems. And that's kind of important because really the spread of the disease is associated with relatively high levels of moisture. And that's why Trinity County comes on the very, very edge of this aerial or range of distribution that's favorable for the organism because it gets a lot hotter, a lot drier in Trinity County. Nonetheless, you do have several parts of the county that are favorable for the organism. One of the interesting things is actually it needs rain to spread. So the organism becomes infectious only during rainfall. And in fact, we have done several studies. We know that we need to have a significant amount of rainfall for the transmission phase to pick up. So if we only have rainfall early in the winter and then it stops, that's not a very good year for the spread. But when we have kind of rainfall, even moderate rainfall happening throughout the winter and the spring season, then those are years that are usually very, very effective for the spread of the disease. It spreads aerially. So it actually produces spores that are carried by the wind, but the spores are gigantic. They're gigantic for the world of microbes, so you still cannot see them with the naked eye, but they're like little elephants, basically. So they're not carried by wind very well. They glide, but they glide at a relatively short distance. And this is important for you guys to kind of understand what is the likelihood of finding the disease in different parts of the county. Normally, they will spread just a few hundred yards in a year. That's kind of the regular distance at which they spread. However, occasionally, if you have a strong storm, or if you have a weather front with a lot of wind, a lot of rainfall, it may spread 1 or 2, or maximum 2.5, 3 miles. So still not huge distances. And it's one of these events, probably in 2011, that brought the organism from Humboldt County to Trinity, basically across the border. So again, that's kind of the range. The reason why we are really interested in understanding the fine scale distribution of the disease is because of the short distance at which the organism moves. So really, if you're in Weaverville, based on what I understand about the distribution of the disease, you don't really have to worry about it because the organism is too far. So simply saying, oh, the organism is present in Trinity County doesn't really help anybody. In fact, where you have oaks, the risk for infection is only when the front of the disease is about 200 yards. So very, very, very fine scale. In the case of tan oaks, we could increase that a little more, maybe to the 1-2 miles I was talking earlier on. So the risk is a little bit broader, but still we are talking about relatively fine scale. So how do you get fine scale of an organism that's spread over about 800 miles? We really don't have the resources. And so the idea came to me 8 years ago to say, hey, why don't we get the local people to just do the work for us? And the idea was presented to some people that are here. And the very first time, the people told me, oh no, it's impossible because people will do a crappy job. And I said, no, I don't really think so. I think people really care, and I think people will do a very good job. And in fact, people did a very good job. And we realized 3 or 4 years later how good of a job people were doing because the data were incredibly strong. And right now, actually, I want to let you know, we just published a peer-reviewed article in a very high visibility journal that shows that the data collected by people like you has produced the best predictive model of where the disease will spread than any other model produced simply by scientists. So this is kind of evidence that these associations between academia and volunteers can really work. Again, there is a trick though. There's 2 tricks. The first trick, I and people that organize these blitzes really make participation in these trainings mandatory each year. So we want to refresh the ideas. We want to show what to look for. We want to remind people what they're supposed to do each year. So we keep the knowledge pretty high up. So when people go out, they know what they're supposed to do. And then the second trick is that UC Berkeley, we test all of your samples. So we're not, we don't take it, oh yeah, we've seen something of death there. We actually, all of the samples get tested in the lab. And so thanks to that testing, the data that is produced is very, very, very solid. And so we can make a lot of inferences based on the data. There is also one peculiarity that I need you to be aware. About 500 to 600 people participate each year throughout the state. But the data you're going to generate is going to make public. And I think 2 years ago we calculated, because we were on the front pages of several newspapers, plus we got 250,000 hits on my website, that we had probably had over 3 million people look at the data. I don't know whether look at the data in an intelligent way or just look at the data. But about 3 million hits. And this is pretty amazing that work of 600 people becomes available to the larger community. And in this case, it was mostly the greater Bay Area. So basically, the data you generate can be used by everybody, and in fact is used by everybody, because since 2 years ago, we decided to put all of your data, plus all of the government data, and all of the researchers' data into a single database, and that database is called the SOD map. And the SOD map is available on the internet, and it's also available through an app that's available for free, both on Google Play and on the App Store. And what the app does will allow you to see when you're out in the field, where you are, in the location of the closest infection center. But it will also do something more that's particularly useful for people living south of here that have oaks. It will tell you what the risk of infection for your oak is, or for an oak that's in the same location as you are. So these are instruments of the web through the SOD map or SOD map mobile. They're instruments that are used to actually look at the distribution of the disease. If the disease gets close to you, then you know that you need to think about management. And it's very important to think about management preventatively, because basically almost everything we know that works needs to happen before the disease arrives or before the disease infects trees. So if we wait for the disease to become established, it's too late. So the disease is called southern oak death because it kills 3 species of oaks. But really, the most important vector for the disease is California bay laurel. And the second most important vector is tan oak. So oaks are kind of an accident. It's an unfortunate accident because oaks die in large numbers, and tan oaks also die in large numbers. But it's not the infection that kills oaks, and it's not the infection that kills tan oaks. Those are infections on the stems that are actually spreading the disease. It's very, sometimes, inconspicuous infection on the leaves of bay laurels and on the leaves of tan oaks that actually spread the disease. And these are not infections that cause significant mortality or even significant death. The ones on tan oaks can cause a little bit of twig dieback, but that's about it. When the spores move from the leaves of these 2 trees on the stem of tan oaks or on the stem of oaks, that's when the deadly infection takes place. So it's a very distinct phenomenon. I also like to point out that the stem of bay laurels doesn't get infected. So bay laurels are not affected by the disease. So there is no inbuilt control for the disease. The disease can be spread more aggressively because bay laurels don't suffer at all because of the infection. So that's kind of unfortunate. And when tan oaks get killed and when oaks get killed, what takes the place of these trees? Despite the old models that predicted other species would take place, it was predicted the Douglas fir would take a big chunk of the space that's left by dying oaks. Instead, it's California bay laurel really takes over significantly if it's present in a site. So the situation just gets a little bit worse. But this is what we'll be looking for. So we're looking for those leaves that carry the infection, because plucking a leaf is relatively easy, and I never said it, but even if it's in private property but the branch comes out, you can actually pluck a leaf and you can put it in a collection envelope. It's legal. And so it actually allows, without harming the tree and without too much effort, to get information about the presence of the disease. And it actually allows us to figure out where the infectious trees are, rather than looking at oaks which are not infectious. So really it's actually even more useful from a standpoint of understanding where and how bad the risk is in any given location. South of here, where you have oaks here too. So you have, I guess, you have canyon-like oak here, which would be infected. Oregon oak does not get infected. The blue oak doesn't get infected. So it's only California coast live oak, California black oak, and Shreve's oak, which are present south of here, that die because of sudden death, and canyon live oak, I guess, which is also here, that can get the infection. Most oaks get their infection from California bay laurels. So the infection is, as I said, sometimes relatively inconspicuous on the leaves of California bay laurels. If you had supervision like I do, you will see this particular little structure here. The structure is a little bit like a pod, like the division of body snatchers. You know, that helps you to kind of connect things. So this is a little pod, and the little pod is airborne. As I said, not very large distances. And the little air pod will land on a plant surface. And guess what happens? There's a hatch at the end of the pod, and the hatch opens, and about 20 spores will come out. They're very tiny spores. They're the aliens inside the spaceship. Each one of those 20 spores will infect potentially, you know, a tree or a leaf. But this process of exiting the pod can only occur if there is a film of water, because the aliens, so the little tiny spores, need to swim. They don't know how to walk. They can only swim. So if you don't have a film of water, basically the swimming process, which is what leads to the infection, doesn't happen. And this, if the film of water doesn't stay, there for several hours, the process is not optimal. It gets interrupted because there's not enough time for swimming. So the organism has devised a mechanism not to waste any energy. And what is the trick? The organism only produces and sends the pods out when it's raining. And it hasn't learned to use fog yet, so we're fairly lucky. But when it's raining, the organism is perfectly timed to start the process of production and then of emission of these structures that are infectious. This, as long as there's a little bit of rainfall, this process happens every year, and this is why we're here now, even if it's a fairly dry year. However, oak infection only happens when we have a way above average rainfall level. And that's because you need a lot of spores to infect an oak. And those basically are only generated when you have a lot of rainfall. Another reason why serving for oaks is not a good idea, because you only get infection relatively seldomly, just once every 3-4 years. But you always get the infection, and you always get a ton of infection as long as you have a little bit of rainfall. So what you look to do today is we provide you with what we call a collection packet. And the collection packet really is everything that you need to collect. And we go through what you'll be looking for, how you're going to do it. So because I know that it's, you're thinking about the basketball game, and it's 6 o'clock and you're tired, so I'm talking, you're probably thinking about something else. So you have the instructions inside. Okay, so in case you're not really listening to me, everything that I say is summarized inside the packet. There is a pencil that you can use to fill in the different forms. There is symptoms page, and if you can return, these are fairly expensive, so we can use them for other surveys that will remind you what to look for when you're out in the field. There is a little bit of flagging tape, and flagging tape came the third year when we realized this old lady, she was amazing. She collected a lot of samples, but she tended to collect samples from the same tree over and over again because she didn't remember that she had sampled that tree. So she would go around and resample the same tree, and then we figured out that she was sampling the same tree. So we decided to give her some flagging tape so that she would know that she had sampled that tree. And so that's why we have this. But inside the packet there's also, and we'll get to that later, we are now providing aluminum tags and aluminum nails. It's a very good idea if you use those, because then that tree will become permanently identified as a tree that's either surveyed or a tree that's sampled. And then the most important thing is there's small envelopes inside here. The small envelopes is where you're going to put your samples, your leaves. Okay, and we'll go through it, the process. And then there's data sheets. So inside each small envelope, you need to fill in a data sheet to go with it. And the data sheet tells us a little bit of what you're doing in that moment, what you're seeing. But more importantly, it tells us where you are. And I'll tell you how you can know where you are. There's different approaches. So let's start by telling you what you'll be looking for. So these are bay leaves, and basically you'll be looking for relatively distinctive lesions that are present on the bay leaf, and the location of the lesion, which basically is this brown or sometimes black tissue, which is dead leaf tissue, matches normally where you would think water accumulates. Because remember again, water is important for the infection. So because most leaves are carried with their tip downwards, but not all of them, most lesions tend to be present on the tip of the leaf, okay? So let's start by discussing this and then we'll move to those. So the lesions are present on the tip of the leaf. The edge of the lesion, so this is what you'll be looking for. The edge of the lesion should not be a straight line. Okay, if it's a straight line, you're looking at something else, okay? So it should be irregular. You see how irregular it is? Because this is a biological, event. And at the edges of the lesion, you should have one or both of two very distinctive traits. One of them is kind of a darker line. I don't know if you can see it here. It's like a black line. And often in association with a black line, there is a yellow halo, okay? So that's kind of these other, and if you're lucky, if you're like in a very moist forest, and you may also see little individual dots. And those individual dots are the location where little droplets remain. So in the morning if you walk through a forest, you will see water accumulates on the tip and then you see little droplets. So in correspondence with the little droplets, you have these little dots, okay, which are smaller infections. Now, because infection occurs where water accumulates, if the leaf is carried sideways, you actually have the same symptom developed over the lower blade of the leaf. So it looks a little bit different because there's more room for the lesion to grow. But as you can see, basically water's been accumulating on the lower blade, and you have this lesion on the lower blade, again with the irregular margin. Often it has what we call a pixelated, like pixels, a pixelated look, but all the same traits apply also on the side of, the sideway lesion. If you find a tree that has leaves carried downwards and leaves carried sideways with these two types of lesions, that's the perfect tree to sample, because you have the highest chance of actually getting sudden or death or a close relative of sudden or death. So I should say that unfortunately the symptoms do not prove that you have sudden or death, because it could be one of many things, okay? But the closer you are to what I'm showing you, the more likely it is that you're actually collecting the right type of material. Sometimes a leaf is very big and it gets heavy with the water, so it's arched, and so you will see a lesion near the petiole, and then you will see a lesion on the tip, okay? So there's all these different patterns that you can actually have. Color of the lesion is relatively important. So the lesion means that the tissue is dead, so you can see the lesion on both sides. So it's not something on top of the leaf. And it's not a powder that covers the leaf, like an unsightly powder that covers the leaf. That's something else. Those are pseudomolds, and they have nothing to do with sudden or death. So don't, if you see kind of an ugly powder on top of the leaf, that's not what we're looking for. We're looking for a leaf that has a dead tip or a dead underside. And you can see the dead tissue on both sides.
So there are other hosts for sudden death. Are you only sampling bay laurel?
And Tanox. These are the two main ones, because the other hosts normally, especially at the beginning of the epidemic, are relatively inconsequential, and they're only infected under an infected bay laurel or under an infected Tanox. So by just focusing on these two hosts, you actually cover all of the other hosts. The only exception would be redwood, but again, we only see redwoods infected in areas where the infestation is being significant for a long period of time.
So only the pink parts on the map?
We'll get to that. So the color of the lesion is important. The best color is actually when the lesion is brown. That's the most common one. However, if the pathogen is very active, you know what happens? This black line here, it produces a black line, then another one, another one, another one, another one. And as the end result of these multiple black lines is that the lesion looks black. Which is interesting because a black lesion, we perceive it as different, but it's actually the same thing. So brown and black is really good. A 1-year-old lesion becomes gray. So gray is not ideal, but if that's all you find, so basically this would be gray, you can still collect it because we can still identify the pathogen. Okay, but it's not really the best type of material. So you want to look for mostly brown lesion, which would be infection of this year or maximum of the end of the previous year. One thing to keep in mind is that, especially in a dry year, it could be as few as one leaf that's infected, okay? So don't expect the entire canopy of the bay laurel to have these spots. It could be one branch, it could be, normally it's not just one leaf, normally it's a little bit more than one leaf, but it could be just a branch with maybe 3-4 leaves. So you really have to pay attention, and I'll tell you what we have figured out works. And we're very lucky that normally the branches infected are in the lower canopy. So normally you can actually reach them with your hands and you can see them clearly because the organism likes the shade. And so normally it tends to prefer these branches that are lower in the canopy. So that's where you should focus. You should focus on the first scalp, the lower canopy of the tree. What we figured out is that in order to feel relatively confident, so the best way, there's a secret, the best way to get rid of sudden of death is to survey like this. Because if you survey any area of California like this, walking, there's no sudden of death. I tell you, so we know how to get rid of it. Because you don't see it, it's impossible. So even an expert by walking, you don't see it because of the models and the light so you actually need to stop and look at each side of the tree for 20 seconds. Some people can be faster, some people can be a little slower, but we figured out that 20 seconds is a good in-between time. So you get to a tree, 20 seconds, 20 seconds, 20 seconds, which is an eternity. But that is really the best way to do it. So if you can look at each side on 20 seconds, for 20 seconds, then at least you can feel relatively comfortable that you didn't, the tree potentially didn't have an infection that was visible. In your case, you probably won't have questions, but you should still do it. You're gonna sample, so each packet allows you to sample from 10 trees. We hope that each of you is gonna take at least 2 packets. It doesn't take that much to find 10 or 20 trees if you're the right type of forest. However, you may look at a lot more trees than 10 and 20, okay? So we'd like you to keep track of how many trees you look at, and then how many trees you actually sampled, we'll know. So how many trees you looked at, and how many trees actually had symptoms. So as you walk, oh yeah, this tree has symptoms, but you just sampled another tree nearby, so you're not gonna sample that tree. But nonetheless, we would like for you to keep track. It doesn't need to be perfect, ballpark is fine, because at the end of the day, we would like you to put 2 numbers here. How many trees you looked at that were healthy, and how many trees you looked at that had symptoms, which doesn't mean that they had sudden death, remember, because the symptom is very generic. But those numbers are important for us to calculate the actual infection rate in your county. So this may be a number that's very, very important. The other host that we would like you to sample is tan oak. And in tan oak, we would like you to collect tan oak leaves. In tan oak, the symptom is completely different from the symptom on the laurel leaves. So in tan oak, the symptom is associated with the mid vein, okay? So what you should see, and I don't have a pointer, but you should see that where the mid vein links with the twig, there's a petiole, I call it a petiole, but somebody told me it's not really a petiole, but it should be green, like in this panel here. This is not sudden of death, okay? Because there's no association of the symptom with the mid vein. But these 2 pictures here are pictures of leaves infected by sudden of death. And at 2 different stages. The first one is kind of an early on stage, where the infection has colonized just the area of the petiole, and the leaf is starting to dry out in weird patterns, because there's no connectivity between the leaf and the rest of the branch. And you can see the browning very clearly. You see how brown it is, close to the petiole? Right there. And this is an infection that's progressed a little bit more. So that mid vein is supposed to be green, and as you can see, it's brown. Okay, so any of these 2, again, the drying pattern is unpredictable because the leaf will dry out depending on a variety of factors. But the browning of the mid vein is what you'll be looking for, and the browning of the petiole is what you'll be looking for. Okay? Here is an example of what not to collect. Okay? So there's an association with the mid vein. If there's an association on the tip, like it would be on the laurel, that's what you're looking for. You want association with the mid vein and the petiole, or the entire mid vein. Okay? So the right hand side is what not to collect. Okay, so let's say that you found a tree that has symptoms. We normally ask that you collect multiple leaves from the same tree. If you can collect 5 leaves, that's really good, because then we look at all of them and we pick the best one for the lab testing, okay? So don't just pick one, pick usually 3 to, if you can pick 5, that's a good number to remember, okay? And you have the small envelopes, you will put all of the leaves inside the small envelope. If you want, you can flag the tree, or you can put the aluminum tag if you can do it and you want to do it, and it's very good if you do it. And then the most important steps is to let us know where the tree is, because if you don't know where the tree is, then really the whole exercise is pointless. So you really need to pay attention on figuring out where you are. And there are 3 ways that you can do this. One way is to use a GPS device, okay? So if you have it, but many people don't have it. If you don't have a GPS device, you have a cell phone and you have a reception where you are, which is the big question. So probably won't work for most of Trinity County. If you use SOD Mobile, it actually, if you tap the risk button in the middle, it gives you the latitude and the longitude. So it's very useful because at the same time you can see where the infection is on the screen, and then you tap the risk button and it gives you the latitude and the longitude. So if you have reception, you download the SODMap mobile app, which is free, then you can use that, and you simply write the longitude and the latitude in the card, and I'll show you exactly where to do that. One thing to remember when you write down latitude and longitude is that you don't round up the numbers, okay? Put all of the numbers, please. All of the numbers you see, just put them all, okay? Because if you round them up, you actually change in the location, so it's not what you do. So, if you don't have a GPS, or your phone is stupid, or you don't have reception, then there is another approach which we found works really well. Basically, you need to write down some information about, that is familiar to you, because hopefully you'll be collecting an area that you know, or you're somewhat familiar with, that's gonna remind you where the particular tree was, okay? So whatever landmark you see, whatever you remember about a particular trail, then when you go home, you need to turn on Google Earth on your computer. If you don't have it, you can download it for free. And then what you're gonna do, you're gonna redo your hike. This is the best that you can do if you don't have a GPS or you don't have reception and then you're using a phone. To turn on Google Earth, and then you basically redo your survey with the mouse, to the best of your knowledge. And then you just let the mouse be where you think the tree is, and at the bottom of Google Earth you will see the latitude and longitude, and you just copy those on the form. So, for instance, let's say that you said, okay, I collected from the small tree across the parking lot. That's what you put down as a note. So the parking lot here is a big tree, is a small tree. So when you're on Google Earth, you actually see the small tree. You don't need to put a place mark. You can simply put the mouse on top of the small tree, and at the bottom of the page, you will have the latitude and the longitude. So this is the second best option. So the first option is to have a GPS device or a phone. If that doesn't work, don't despair. Just try to remember the locations, and then try to redo your survey with the mouse. And then let us know what Google Earth says. Now, one of the things is, if you put the little data sheet inside the envelope and you're sitting at your computer, the first time I did this, guess what was under my chair at the end of my little second survey on the computer? A lot of leaves, because as I was pulling out the data sheet, the leaves were falling out as well. So be careful, because that's something that definitely can happen. If these two options are not available, and you are in a relatively small property, then you can give us the address and an approximate distance from the address. So it could be 200 yards southeast of 20 Wisteria Lane, okay, or whatever you want. So you can do that. That's really the least favorable option, because it's going to lead to a lot of mistakes, and probably here we're not talking about small properties anyway. And then if you're sampling multiple trees, you can refer the distance between tree, you can say tree number 1 was 200 yards northeast of this address, and tree number 2 was 50 yards east of tree number 1, and so forth. So you don't have to refer to the original tree. So, how do you go about sampling? So at the end of the talk, we'll actually show you where the sudden of death habitat is, which is fairly, where you have tannocks and where you have bay moles, basically. That's really where the disease can become established. So that's really where you're supposed to look for the disease. But when you're in a site, how do you sample? What are the best strategies? And there is not just one perfect strategy. It really depends on you guys, what you want to do, your effort, the size of the area that you want to survey. Normally, to identify new infestations, we ask that people identify the symptoms that I've shown you. They collect from symptomatic plants. And normally we ask that people don't collect, if you find a tree that has the symptoms here, and then you find one here, 2 feet away, that is not really, that second tree doesn't really increase our knowledge very much. So in general, we like for people to sample about 50 to 100 yards, one tree from the other. However, there's no fixed rule, because imagine you find a tree that has symptoms and the symptoms are okay, and then 10 years later, you find a tree that has the perfect symptoms. Go ahead and sample that tree as well, okay? So don't give up on that tree simply because Mateo said 50 to 100 yards. Okay, this is kind of just a general rule. If you have a pocket, so let's say that you go in an area that you already know has sudden of death, and you actually want to figure out how much sudden of death there is, then the frequency of the sample can be 5 to 10 yards, okay? So you can actually sample a lot of trees close to one another, and then we'll see how many of them with symptoms, and we'll see how many of those trees were actually infected. And so that will allow us to figure out how bad the disease is in a certain area. If you think that you're going to do this, you should get a few more packets, because you go through them very fast, and then just return the ones that you don't use, okay? So the strategy does require more material, because you'll be sampling more trees that are next to one another. To identify the infestations, again, I gave you a distance, but you really all you have to do is find the symptoms. You can use the SOD map when you go home to see where the disease is in Trinity County, but we'll actually show to you at the end of the talk. Or you can even use SOD map mobile in the field. So this is what the SOD map looks like, just to make it clear. It's very simple. As you turn on the SOD map, this is what you're going to see. This is enlarged. You will see all of California with little colorful icons, and the icons are green or red. A colorful little tree on the screen means that that particular tree was sampled. And if it's green, it means that it doesn't have sudden of death, and if it's red, it means that it has sudden of death. This is helpful because this is from the Bay Area. It helps figuring out areas that have been sampled a lot and that already we know have the disease. Areas that have been sampled a lot, but the disease wasn't there at the time of sampling, so it still may be interesting to go. And then you can identify areas with no sampling at all. Okay? In your case, most of the areas will be like this because you're the first ones. So you're setting kind of the pace and the baseline for the information. And again, SOD map mobile. I'm not even going to go through this, but you can actually use the risk rating to know whether you should prioritize your sample, because if the risk is high in a location, so you tap the button and it says the risk is high, it means that we already know the disease is there. So that's not the first priority for sampling. But in your case, again, you don't necessarily have to worry about that. Okay, so if you know that the disease is in a certain area, you want to determine how bad it is, then you can use these different schemes where you sample a tree every 5 to 10 yards. You can use a linear pattern, or you can use a cross, and so forth, and that can be used to determine intensity or severity of the disease. There is a third option. Ryan and I discussed this a little bit. I don't know whether people are interested, but you should be aware. You can actually sample in transects, which means following a line and sample at regular intervals. And if you cover your property, reserve your property well enough, and all the samples are negative, you can actually get a free from certification, just in case there's anything that goes on in the property. If you've done the survey, then you can actually potentially use the data to show the authorities that the property is free from the pathogen, and so that you don't necessarily have the burden of some, not all of the burdens of the regulation. And normally the way to do is to sample 3 lines parallel, so each one of these lines is a transect, and the transect should cover most of your property. So depending on how large the property is, you can space out the sampling areas differently. So you can do 6-10 points per transect, and the distance between each point of the transect. So a transect, you start walking, and you're gonna sample here, and then you can sample at regular intervals, but the size of the interval can change between 50 and 200 yards, depending on how much area you have to cover. So those are relatively reasonable. If you go over 200 yards, then the survey becomes not intensive enough to prove that you don't have the disease, okay? So that's the very upper margin. And 50 meters would be an ideal one. And then the distance between the 3 lines that you're gonna walk should be also 50 to 200 meters. So basically, you're gonna generate a little grid on the property where at regular intervals you're gonna sample. If you don't see any symptoms within, you reach your sampling point, which is every 50 yards. You look around about 10 yards from where you are. If you don't see any symptoms, you can put down that in that particular location, you didn't see any symptoms, and so forth, you go on. Do not collect plant material that's asymptomatic, because then people in the lab go crazy looking for the little spot that doesn't exist, okay? So just write down that you surveyed, but you didn't see any symptomatic material. Okay, so.
You got a question there, Mateo.
So, sorry.
So that transect on your property, that's assuming that you've got bay laurel and or tano.
Right.
And if you don't have them.
Well, if you don't have them, you're not going to have the disease, because as far as we know right now, I mean, again, the only exception would be redwoods. We have one redwood tree that's infected in the Presidio National Park. We don't know, but again, it's an area of really high, the marine is super infested. So, I mean, those really would be. Then if your bays and your tanox are infected, you may get an infection on every species that grows under them. But that's kind of, it's a secondary.
Right.
But the list of host plants had big leaf maple,
Yeah.
rhododendron, and a lot of other host plants.
Yes. So all
See those?
the species we only at this point, if we had a time machine, we went back to the 80s, you would find maybe some rhododendrons infected, and still we can find some rhododendrons that are infected. But the native rhododendrons are not very susceptible, and the ones that are planted, since the 80s, now are relatively clean because they go through inspections. So now in California, the disease is mostly driven by bay laurel and by tanoc. Once you have it established on bay laurel and tanoc, all the plants growing under bay laurel and tanocs can become infected. So the list of species is very large because there's a lot of species that are growing together with these trees. But if you don't have the bay laurel and you don't have the tanocs, the other species are not infected, with the exception of redwood, and maybe potentially poison oak. So
who's going to sample of poison oak?
you get. So this we're very lucky that this is out the dynamics of the disease, because rather than focusing on 125 hosts or what many we have now, I forgot. We can focus on these 2 because these are the 2 major hosts that drive the disease. And in fact, in areas that are more urban, people are taking out their rhododendrons, and there's no need to take out the rhododendrons, because if you don't have bay laurels that carry the disease to where the rhododendron is, the rhododendron doesn't become infected. So it's a two-step process. The tanoc and the bay laurels will take the disease to a location, and then once it's a location, will spread on many other hosts. But the best way to identify the disease is to use these 2 hosts that are the most susceptible.
Was there a question over there? Any other questions?
Yeah, if your property is void of tan oak and bay laurel, can you get a free from certificate, or do you still have to do transects, or? Because right now, all of Trinity County is under the ZOI, and there are
Yeah, I
areas
wouldn't-
where
yeah.
it's nowhere.
If you don't have any of, unless you have them right on the border of the property, then that's a different issue. that you may be required to do some testing to make sure that you don't have, the pathogen on some of the other hosts. So that would be the exception, but if you don't have those hosts within, a certain distance from your property, which I would say 200 yards or so, you probably really don't have to worry.
I'm going to talk about the zone of infestation and the quarantine and what those things actually mean and what's being done. I'm sure you kind of a couple minutes after this gets done.
So the reason for the tagging is, the tag on the tree is really good because then we can really identify the tree that you sampled. And let's imagine that the tree comes out to be positive and it's the only tree to be positive in a certain area. Potentially you could also think about removal of that tree. There's a lot of things, but if you can tag the tree, meaning that you have, you have permission or it's an area where you can be tagged and so forth. I mean, we give you the tags. They're aluminum tags, they're aluminum nails. So it's safe to tag the tree. And for the whole project, we decided to pick the northern side of the tree and about 1.5 meters, about breast height, for the tree. So we all using the same tags. The tags are also important if you're sampling a tree in an area that is free from the disease, and you you pick these trees well. These are trees that are pretty large and they may catch the disease, because you can use these trees as sentinels, as monitors. So basically you can go back every year to these trees and keep checking whether the disease has arrived or has not arrived. And so if you have a tag there, then you'll know that the tag trees are your sentinel trees. They're your monitoring trees.
But you said not to collect leaves if it's asymptomatic.
Yeah, so if it's asymptomatic, you don't collect the leaf, but if you did a good job with your 20 seconds, then you can write on the form, no symptoms, okay? And in fact, there's pink forms that can be used for trees that don't have any symptoms. And in areas where there's a lot of disease, normally people use the pink forms, and they just write. In this case, you can use also the white ones and just write no symptoms and give us the location of the tree. And we'll go through the form in a second. So if you have symptoms, please use one of the white labels. Each little label should go inside the envelope where you're going to put multiple leaves, right? So you're going to put multiple leaves and then you're going to fill in the label using a pencil. Don't use a pen because if it gets wet, then it's a mess. So the pencil is better. We give you the pencil. Thanks to Funding for the Fire Service. So let's go through what you need to fill in. If you don't fill in all these fields, we actually toss the sample. Okay, so we really need you to pay attention. And there is a reason for each one of these fields, and I'll show you why. I should also point out, this is where your time is incredibly valuable, but each sample costs a lot of money to process. So it's not, so basically we really need to use the money on samples that were collected. So we need a collector name or initials. Okay, so if you participated in the survey, we need to know your two initials, or you can put your name, you can put your full name, we don't care. But we need to know who you are so that you can actually link your samples, because there's thousands and thousands of samples in the database. And you'll never be able to find your samples unless you know, and I'll explain to you how you do that. But also because often what happens when we put the samples in the result map, we make mistakes, because maybe you made a mistake or we made a mistake in reading a number. So then you go back to your samples and then you let us know, hey Matteo, these samples are in the wrong place, and you tell us where to put them. So it's been a very valuable two-way process to perfect the maps. And so please do that. Your first tree, the first tree you sample or you survey is your tree number 1. And then your second one will be tree number 2 and so forth. So that's how you do it. The date, then you tell us whether it's a bay or a tan oak, but we could figure it out. If you put a metal tree tag, then put the number. That's really important. So we need to know the tag number is associated with that particular tree, which then we know is in a particular location. Tell us where you are. So private land, tell us whether you are in public land, which would be, for instance, Caltrans, so the side of a road, it would be public land, or if you are in a park or open space, designated so. So if you are in a national park, or in a state park, or a county park, or so forth. If there is a dead oak or a dead tan oak, visible, and not like on top of the mountains, like within, a few yards from you, just tick that box. And remember, we will not know, and you will not know why that oak is dead. Maybe it's dead for a completely different reason. Nonetheless, it's data that we are putting in the database, okay? But again, we won't know whether it's sudden or death. And then here, this is where, the location, this is where you can write down some information for you or an address if you're gonna use the address route. But remember that the most important thing is for you to tell us the latitude and the longitude, either by using a GPS device or a smartphone or by doing the Google Earth little walk on your computer screen, okay? So that's, the symptoms that I was talking about are a dead oak or a dead tan oak. And there is something peculiar that most oaks and a certain number of tan oaks that are infected by sudden or death is, and it's the presence of this bleeding of a viscous sap, brown or black, that bleeds from the bark, but the bark is intact. So the bark is perfect. There's no wound, there's no insect holes, there's nothing. And you have this viscous bleeding outside the bark. And this is very different from and it smells relatively good to most people. It smells like wine fermentation, but it's a good smell. It's very different from the bacterial wetwood that many trees have when there is some kind of a breakage on a crotch or a branch is a little broken or there is decay on the stem. Sometimes you have all this black stuff that oozes out. It's very liquid and it smells really foul. That's not what the sudden or death symptom is. Again, it's difficult to know for sure whether it's, but if you see something similar to this, of this droplets and it's kind of viscous and it smells pretty good, that's a symptom. But mostly you'll be looking for dead oaks and dead tan oaks in the vicinity. You have 2 pink labels without a small envelope, so that's to tell us location of trees that were healthy. But in this case, because you don't have a lot of disease, you can also use the white ones. So again, if you're doing a survey and you keep not finding anything, but you want to let us know that you're not finding anything, then just write down no symptoms and tell us the location. But the rest of the information has to be there to be entered in the database. And again, that's okay. So then at the very end of the day, when all is said and done, we need you to put the same initial that you used, on also the registration, the sign-up sheets with your email, because that's how we start the communication process. Then we ask you to tell us ballpark, or precisely, some people are very anal and some people are not. How many trees did you actually look at? Besides the one you're telling us in the survey. So you may tell us about 10 trees, or you may collect from 10 trees, but you may have looked at 100. So let us know it. Out of the trees that you looked at, how many trees had symptoms and how many trees were totally clean of symptoms. So just let us know. And again, don't go crazy, just give us an idea of that. So once that's done, you will have to return so all your samples have to be inside the envelopes. All the envelopes have to be inside the packet. The packet needs to be closed and sealed, as well as you can. And this needs to be returned in some collection bins, and Ryan will explain where the collection bins are. And he will also talk about dates. So really, I think there's 2 weekends that we have selected for Trinity County. He will explain to you. So basically, you cannot collect, let's say, tomorrow and then return the sample a week later, okay? So really, you need to collect, and then within a day, you need to return the sample to the bin or so. So from the moment you pluck the leaf to the moment you return the sample to the bin, it has to be only a day or so. And also, if the sample stays in the bin for a week, that's not good either. So that's why we have to have dates, because then we come and pick up the bins with all the samples. And Ryan will talk about about the options for returning the samples. But normally, we would have you guys go and do your survey tomorrow and the day after, and then return by 10 a.m. on Monday or 8 a.m. on Monday. I don't know, each area is different, but that's normally how it works. So now, the thing that could really ruin it for all of us is you do a beautiful job, you collected perfectly symptomatic leaves, and then you leave this, you go and swim in the lake and you leave this on the dashboard of your car. because this will fry the sample. So the most important thing, you don't need to put it in the fridge, but it needs to be kept in the shade at all times. Especially cars are notorious for killing, samples. So try to keep it in a cool spot. Do not expose it to the sunlight. Please do not put it in the freezer. Because while freezing, it's okay. When we defrost it, it turns into a mess, and then we actually lose the information. And when Ryan will tell you, whatever you guys decide for the deadlines, I used to have people that, if the bean is gonna be collected at 10 a.m. and you arrive at 11 a.m., then that's, really doesn't work, because that bean is already en route to Berkeley. And we're doing the entire state, so basically, you need to return the samples on time. So again, where to sample? This is a little bit, this applies to the entire state. So basically, wherever you have bays and tannins, okay? That's kind of the most important piece of information. If you have patchy forests, which is not the case, at least here, from what I have seen, normally the organism doesn't like small patches. It prefers larger patches and the continuous forest. So that's really where you find it. The 2 or 3 tips that I have to give, actually instructions. If you're going to an area that may have sudden death, these leaves that you're collecting are very, very infectious, okay? The infection will not be transmitted to humans or animals, but it can be transmitted to other plants. So if you're careless, so if you pick some leaves and you throw some in your car and some in the small envelopes, the ones that are outside the envelopes can actually transfer the disease, okay? So instead of helping, you would be not helping. So make sure you just put the good leaves in the small envelope and then throw the other ones in the site where you're collecting them. That's very important. And then I also have to tell you that if you go to a site that's very wet, maybe even if it doesn't rain because there's a stream and your shoes gets covered with mud, and you're actually targeting areas that may have sudden death, you may have a little bit of sudden death in your mud. If the soil is dry, there is almost no risk, but if the soil is very wet, there's a little bit of risk. So normally we recommend that you clean your shoes afterwards before you exit the site as much as possible, or you can clean them in a sink that goes into a city sewer system. Don't clean a sink that goes into your yard, because otherwise you're just putting the pathogen in your yard. So the chances of moving with the soil from just shoes, it's not very high, but nonetheless, it's actually there. Okay, so now we're going to show you some maps, and you guys are going to talk. First of all, I wanted to thank Ryan, who's a local organizer. So we have 21 blitzes, I think, this year throughout the state, and they all happen because there's one local person that puts it together and organizes it. And then I just show up, or some of the people from my lab who just do this. So really, this is a cooperation with local people. And then I have to thank the Forest Service, State and Private Forestry, which is the main supporter of the activity. So they pay for most of the lab analysis of the samples that you collect. And without that funding, there would be no SOD Blitzes. So these are the URLs that I showed you at the beginning. So here they are again if you're interested in looking at them. What I wanted to show you, and then Ryan can take over, is this. This is an animation, maybe a little dated because it's from last year. It's an animation of what we predict the potential spread of the disease, and high risk is identified actually by a red shade that will show up on the map. So, and yellow, it's low risk. So you can see, this is actually historical, so it goes back a few years. So you see 2015, so which is right now, you can see areas that, and I think maybe the other maps will show it better, but you can see the areas that are at a low risk. And then as you move on, you can actually see the areas that are high risk, which are the red colored areas. And that's 2030. So again, you see kind of most of the red is here, that I guess mostly it's in this area here, where we already know the sudden death has arrived. So this is kind of a prediction model, but remember where, it's, the issue is where the hosts are, especially Beloro and Tanuk, and the second issue is also where is disease already present, because that's gonna be the most likely source. And so if you're gonna go where you already know, or we already know, and we can show you on the map where the disease is, remember that the chances are the sudden death may have already have arrived up to about 2 miles from that location. And that would be very good information to have. Okay, so if you sample the entire area 2 miles from where we have established infestations, are areas very likely to already have the disease. Not necessarily, but and then as you move further than 2 miles from the source, it becomes less likely, but again, not impossible, because movement is not always linear. It doesn't always follow a precise pathway. And sometimes you have multiple independent movements of the organism following unpredictable and unforeseeable pathways.
I was just curious if you see any more movement along travel corridors, like here we have from infected areas and from in the coastal regions through Trinity. Is there more?
So this is a very good question. There is a proven, because it's published, so some soil analysis have been done, association between waterways and the disease. And it doesn't seem that it's not because the water is actually carrying the disease, but because the conditions are favorable. Sometimes you have wind tunnels along waterways and it's moist. So definitely riparian corridors should be a #1 priority. The evidence about roads is inconclusive. I personally think that there is an association, but people that have done, structured studies have not been able to prove it yet. So it doesn't mean that it's not there, but there is no strong evidence. But there is some good evidence that ridge tops and draws definitely get the infection early on. So, and the pattern is, normally the biggest trees are like catalysts. They're they're the ones that catch the disease, and then most of the movement will occur using the lower limbs, but the original catch, the original tree, usually it's a big tree on the ridge top, and then it will move sometimes from the ridge top following draws. So ridge top with big tanuks and big bay laurels and riparian areas, those should be a priority. And then potentially, it's not very likely that it's going to be in a very remote area if it's far away from trees that are already infected, if there's no people going there. Okay, so again, some kind of roadways and assisted, that's still something that I would keep in your mind as I'm still going to keep looking, even if the evidence was not really conclusive.
So I had kind of a two-part question about
Yes.
the soils. So, so if there's a risk of transmitting the disease in your soil from your boots or something where you collected it, what about, the issue of all the soil that comes in from the dope growing areas that goes to all different parts of that county? Is there a testing program for the soil? And then would it be good to have some sentinel reference trees around the big dope grows or something to see if they're like a infestation point?
Well, first of all, I'm not sure that the dope growing areas are necessarily hotspots of cellular death. It doesn't.
But do you know? Have you tested?
No. Okay. So there's soil becomes infected, but we have very few cases where we've been able to show that the soil was actually the pathway that the disease used, and mostly because the disease does a very good job of moving through the air. And we know that foliage really, mostly it's foliage that's moved around, that's infected, that potentially has a higher chance of spreading the disease. There's a couple of cases where we have thought maybe soil could have been involved, but we couldn't really prove it. So in my mind, the soil issue is, it's kind of an intermediate risk compared to what the organism does by itself using the air. But it's also true that through the air, it moves a relatively short distance. And if you're moving soil long distance, then you are moving the pathogen from location A to location B, which could be 100 miles away. So there is a risk. And then it may take time for the organism to come out of the soil and infect the plants, but it's all been shown that it's theoretically possible. So all I'm saying is, yes, it's a pathway. Is it tested really well? No, I don't think it's tested very well because mostly we're focusing on infected plant material because we know for sure that that's the best way for the disease to move around. Nonetheless, we really tell people that, are responsible for movement of soil, especially if there's large movement of soil, like with, PG&E or road building, that, to do their best not to move soil from areas that are infected to areas that are not infected.
And a lot of the long distance spread of the pathogen, where it has actually established itself in new areas, appears to have been from live trees, transport of live plant material.
Yeah, so live plants is absolutely the best way to spread the disease.
So, I was talking to one of the horticulturist that teaches plant technology, and she was discussing unsterilized soil units from chipped forest products that might have been forest products.
Yeah, no, yeah, that's true. That's why, for instance, mulch that comes from infested areas, you should really use it. So the risk is never zero. It's never zero. However, this particular pathogen belongs to a group of it's a Phytophthora, which is a genus that comprises almost the entire genus has pathogens. This particular group is really evolved to be a very good aerial pathogen. So the soil populations, they are alive, but the soil populations that are not, there's other groups, other Phytophthora that have adapted to be really good soil pathogens. This is a little bit different. It's now that we are understanding more and more of this, so there's a couple of papers that we published about how infectious is the soil. Yes, it's infectious, but it doesn't actually, it's not all that infectious compared to any live plants. While other Phytophthora, the soil is very, very infectious, at all times. So again, in terms of nothing is yes or no, it's always a gray zone. But within the gray zone, the soil happens to be, at kind of a lower level. I cannot tell you the word about it, yes, because you could move it, but I can tell you that's not the primary mechanism that the organism used to move around.
But if you're like soil building using infected tissue by breaking it down for non-mineralized amendments to soil that can be brought in, by, like she was saying earlier, by people growing, vegetables or crops or something.
Yeah. So if you embed infected plant material in the soil and then you use that in an area that from an area with the disease, then you're going to bring the organism, and then eventually the organism is going to come out of the soil and infect a plant. But we're still in a stage where we haven't really seen that. We know it can happen because we can do it, artificially. So we know it can happen, but we haven't really quite seen it yet. And so far, because here we're about early detection, it's for early detection. Basically I'm telling you, clean your shoes so that you're not, in case so we're sure we're not spreading it. But for early detection, all of the examples of early detection about sudden death have come from plants, from infected plants.
So I have a question. I brought, years back, brought some buckeye trees from Tamales, where Tamales is, to here.
No, it's not very likely because buckeye gets infected again, but it gets infected really rarely, and usually it's infected really, it has to be really close to bay laurels, and it doesn't seem to be very infectious. So again, it's never zero the risk,
Right.
but in this, like for human health, it's all an issue of likelihood, and so it would be relatively low on the scale.
Seems to me like in the areas particularly where the most susceptible are, we're going to not have cell coverage, and we're not going to have addresses. And I think the Google Earth would work pretty well in most of those areas if somebody has the access to do that. We could use something like township section and range to help find it, or, are there other ways of like assessor numbers or something like that we could use that would help with knowing where you're at, just as an alternative to what?
Well, I mean, we have really brainstormed on how to do this, and the 3 ways that I have explained are the only ways that we can think of. But then really, once you're on site, you gotta figure out what landmarks and what, term of reference. These are the only 3 possible ways.
As good as Google Earth is, I think most of us, if we know where we're at, it should work pretty well. But the cell phones are generally not gonna work.
Yeah, well, that's why,
And we don't have physical addresses.
that's why all the 3 options are all valid. I put them in that order because if you have a GPS, that's always better. So it's the error that we're gonna make becomes increasingly larger when we go from strategy 1 to strategy 3, but they're all accepted because we understand that, this is a real world situation. And we do like the Google Earth one. We came up with that, I don't know, a few years ago, and we tested ourselves. And what we were doing, we were actually putting trees, based on the simple description. So we're asking people to tell us. And we were trying to figure out, and people were saying, 100 meters from the stone where I kissed my girlfriend when I was like 12. We're like, what? So we couldn't really figure it out. But
You can see single trees easily on Google Earth.
and you can see single
It's not in
trees.
the middle, but if it's in the middle of the forest, it's.
There's some level of error, okay, but we're willing to accept that error because there's no other way around.
I've noticed also when you use your Google via your laptop and you have your GPS on your iPhone, for instance, if you know exactly, pinpointed a tree, and you stand under with your GPS on your iPhone and you look at the latitude, longitude, in degrees, minutes, and seconds, well, if you do the same thing on your Google,
It's different.
there's a discrepancy.
Yeah, there's
And
always.
sometimes it can be as much as 10 meters off.
Yeah, the 10 meters, for what we're doing, 10 meters is fine. Actually, I think the Google Earth is actually more precise because it doesn't really relate on the satellites that are available. So if you actually know where a tree is and use Google Earth, now that, we keep fixing some, I think it's actually better. But, as the forest becomes closed canopy, to identify individual trees becomes more and more difficult. So at that point, if you have, a handheld device, it may be better. So, why don't you want to talk about the areas and then keep your questions and then we'll keep answering them. And then remember to, if you can take 2 packets each if you think you're going to do it. And then if you don't use one, just return it at the end, when you return your sample. So no harm done. If you don't have the packet that you can call it back, you get into the mother lode of sudden death and then you wish you had another packet.
Okay, I think a couple important things to bring up are that it seems like some of the common knowledge about sudden oak death has changed and is continuing to change over the past year or 2. So November 2013, they found the one spot in Trinity County with sudden oak death where it routinely gets over 100 degrees, it's very dry. So they're not always going to be locations that are moist and cooler like in some of the coastal counties. There's also some evidence from Humboldt County that you don't need bay, that it can basically spread only by tan oak. Mateo, I don't know if you share those same thoughts, but some evidence from Humboldt County suggests that there are slightly different things we're seeing with sudden oak death for the past couple of years. So the soil movement is a possibility, like Mateo's saying, we don't have any evidence that it's a huge possibility, but it's certainly still a possibility. So Trinity County under 2 different zones of CAL FIRE jurisdiction. This one part right, I think this is, it's Forest District. This line right here is the Coast District. This is under, basically the zone infestation is basically under more restriction than the rest of the county. The rest of the county is also under quarantine, but where regulations are actually imposed, the way that I look at it is really just this tiny sliver on the western side of the county. So Right now, you may not see any effect of sudden oak death regulations in most of the county. It's possible, you can see from the host distribution, there's plenty of distribution across that line that you start to see regulations starting to be enforced throughout the entire county. So it's important to keep tabs on where sudden oak death is in the county, so we can kind of keep the regulations west of that line as long as possible. So let's see, I think that was it for. So just out of curiosity, is there anybody here that is from the western side of Trinity County? Okay, whereabouts?
Southwestern corner, just practically the triangle.
Okay, and there's another?
Yeah, right down at the bottom there, just further down towards the Mendocino.
Okay, so I think logically the first places that we should be looking are in these areas. Unfortunately, if you look at Google Earth, you can see a lot of grow operations right around that point right there. So there's certainly some safety considerations when we're looking at some of these points on here. But I think if there are any spots kind of east of Mad River or east of Willow Creek into Trinity County that people are somewhat near, those would also be good places to sample. You know, it seems reasonable to be looking across the county if there is the possibility of soil transporting sudden oak death. If most people are over eastern side of the county, then you know, it will be helpful at least to say that, okay, it hasn't jumped across the county, and we know it's still kind of in this area, at least from the samples that we've collected. We don't have the manpower to be able to get, you know, a piece of paper that says, okay, the entire county is free of the infestation, but at least we can say in certain areas where we've already looked. So,
But you only want bay laurel and tan oak in the areas where there's a low probability.
right.
So are there any? I mean, I'm pretty sure there's no bay laurel.
No, that's just
And
it.
I don't know if there's any oak up by the lake or something, do you know? No? It's the only place I can think of might have been.
Now that's why it's really important to just figure out where town can be, because that's where you should be looking.
And I think part of this is going to be, I mean, obviously Forest Service has quite a bit of property here that it would be kind of a logical place where sudden oak death would be spreading. Shasta Trinity and Six Rivers is already engaged in a sampling protocol with Humboldt County for this infestation right here. So they kind of keep tabs on this area, but we don't really have anything looking at this area up here right now. So we can kind of discuss if people have ideas where they might want to sample, where they know there are tan oaks or bays. This is just that southwestern part of Trinity County. So this is basically 5 or 6 miles northwest of Xenia, just on the county line, is the only infestation found so far.
So on this map, it's the spread risk map where it's got like moderate risk on the east side of the county. Those are still places where there's no tan or bay laurel. So why are those moderate spread risk areas?
I think that's a good question. Why does the Mietemeyer model show that? There's a couple little places kind of southeast of Weirville. Other than that, see all the yellow in here.
Or going up to the forth from Trinity Lake, you know, there's a yellow patch, but there's no bay laurel or tan oak up to this forest.
Yeah, because I think what it does, the model is based on climate and availability of hosts. And so what it does is when both are present, then the risk goes high. And I think there's no way of taking the climate out easily, so it still shows, it shows areas with moderate risk because climatically they are conducive. So it's, you know, I'm not responsible for these models, but that's probably going to be a limitation of the model because I know that that's how they generate them.
Plus, the scale is really small, I mean, or big, and so those little dots could be bigger if you were to zoom in to like a 120.
And bottom line is, Metemeyer model does tend to overstate.
Yeah, I'm pretty familiar with the eastern border of the county. There's a lot of white oak and black oak, but no tan oak, and certainly no laurel.
I've seen a fair amount of tan oak, but it's in the brush form.
Yeah.
Yeah, that's also susceptible, so.
That's- Humboldt County's found that it is very susceptible as well.
Both the tree form and the shrub form, you should, I mean, it's potential, you know, area. And the thing is, even, you know, the importance of starting the surveys before the disease arrives is that you at least have baseline data. And that usually improves the ability to detect once it arrives. So, but again, your time would be well spent if you go to areas where the disease can become established, rather than, you know, sampling areas where the disease cannot, because.
Stupid layman question, could you show a picture of a bay laurel in California, and a tan oak?
You have a picture here.
Well,
Effectively, it's the least, right?
yeah, it's hard, you know.
Think of the laurel that you know, the bay leaves that you get in the
That one I
grocery
could
store.
probably do, but tan oak, I don't have any idea.
I try to think.
You had a picture at the first, or this one was bay laurel and this one was tan oak, and I
Yeah, but they were-
suspect both of those were Douglas fir.
No, they were just symbols, they're not. So tan oak has, you know, is unique. It's not a true oak, but it's unique because it has a really cone shape. Almost looks like a conifer. It's very cone shaped in its adult form. And I've shown you the leaves, right, for the symptoms. So the leaves are like that. And it does have it does kind of a hairy acorn, so it produces acorns. The leaves can be very different in size.
But oak shaped.
Yeah.
No, excellent.
You saw the picture of the disease leaf? If veins are very strong, it kind of bulges in between the veins a little bit, but the edge is not really serrated. And it's fuzzy behind, it has a fuzz.
Yeah, normally I bring 2 branches, but we're not doing.
Sometimes it's the small multiple trunk trees, and I have some that are 90 feet tall, single trunk.
So bay laurel is very easy. Bay laurel is a very waxy leaf, you know, like the ones that I show you. And if you crush it, it's really aromatic. So,
The other thing about the tan oak is it's evergreen.
Okay.
It's not deciduous, it's evergreen.
And neither of these are in
so I don't tell them when the bees will be.
Yeah.
The times.
Okay, so what we need to kind of set up then is if people are interested in participating in a sudden of death blitz, we need to kind of figure out where people can sample. Once you take a sample, one of the handouts over here has kind of 6 options for drop-off location for your sample. So I guess, does anybody think that they would use a big bar, a big flat, or a Mad River location? Okay.
Big barn.
Big bar?
Mad River.
Big bar, Mad River. And Weaverville, would anybody drop off? Okay, and Hayfork?
Most likely, yeah.
And North Trinity Lake area?
Well, there's no use in sampling up there at all.
Well,
There's Dano present.
You know?
Yeah, there's Dano present, brush form all throughout the entire county.
Do we have access to the internet to show a brush farm kind of here? Is there, do we have
It's
access
just
to
like
Google?
a,
That we
it's
could
just
pull
like
it up?
a full-on
So if it's on acorns, but it's not.
So the important thing is to know where, so these are the beans, I think, the green ones. So, I don't know if you see them. Okay, so you actually really have to tell them exactly where the beans will be because, so that people will find them easily. And then the second issue is gonna be the date. When you, I think
Right, and
you
the
were
dates
thinking
are-
of 2 pickup dates. Are you still gonna do 2 pickup dates?
So, so no longer than 3 days after sampling, you would want them back in here?
Actually, I would,
You would want them being shipped to your lab?
I would say that, within collection, they should be in the bin within 2 days, so that then they can get to us within 4 days.
So I guess what I was planning to do is to come back to Trinity County and collect the samples on Monday of next week. Does anybody, this coming Monday, does,
Following Monday.
well, so do you prefer to sample next weekend or this
That would work.
weekend?
I'm not even gonna go home until Monday.
We can do this both weekends too. If some people think they can drop off this weekend, then I'll come back and collect them.
I think it's usually best to do the collections right after you heard the talk, because you remember all the details. So I think that the idea was that we would give this weekend and then another weekend to capture people that cannot. do
Okay,
this
so-
weekend. It only take, if you know where to go, take, besides the travel time, in 2 hours.
So are there people that think they would collect this weekend? Okay, so we should do both weekends. And did you think that you might take samples near Trinity Lake, or you were just?
I can take samples anywhere.
Okay.
In general area, and I will just take samples this weekend.
Is there an option of sending the samples directly to the lab ourselves? I just need a few weeks in advance to plan my.
It's not possible because we do this throughout the state. I mean, if you're a larger state, if you're like Sierra Pacific, we let you do it. So if you're sending us 100, 200 samples,
Yeah, I work for Forest.
well, yeah, so if you want to send us a large number of samples, yes, we can do it. If it's, just one packet, it becomes unmanageable.
So you want to leave your mailing address and all that? So you-
Well, but the best thing would be, maybe you can communicate with him and he communicates with me. So, but really what we prefer, is that, so if you pick, if you say this Monday you will pick up the beans, the material in the beans, and then give them another, you guys pick another weekend. Remember that you have to collect just the day, the day before you leave the sample. So don't collect the week before they leave the samples because they may dry out. So right, so you need to pick the 2 dates and then, so that people know, because otherwise
Right.
people may collect and gets wasted, it's a pity.
So I guess I'm going to be collecting these bins this coming Monday and a week from Monday. I put
What time?
times for each of these locations on when I'll be picking them up on the handout. It's basically, I guess Weaverville would kind of be the last stop, so I have it as 4 p.m. on there, and then the other sites are mostly 2 p.m. with the exception of Big Bar because the post office, I believe, closes at noon. That's right.
I don't know, actually, I work in that region, so it's like.
Okay. So if you can get the samples to those locations by those times, I'm gonna put them all together and ship them back to Mateo's lab overnight the next day, which I would do Tuesday of next week and Tuesday of the week following.
Does that work for people? Is that for most people? Does not always work for everybody, but.
So you're going to ranch at 4 p.m. on Monday?
Yes. Yep.
Okay.
Were you saying that the Xenia area is pretty well covered by 6 rivers and we don't really need to be sampling there? Or we should actually go down there, that would be the highest priority?
I mean, if you want to do it, it would be good to have a positive control.
If you want to do it, by all means, it would be great to have some samples from there. I know that Humboldt County UC Cooperative Extension Office is planning to go back there in a couple months, but to that specific location. So if you go around Xenia and you're not at that specific place, then if you find it, we can actually confirm things that may not come up in their sampling in a couple months. So if you have the interest, then definitely, if not in that general area, it can be done in a couple months.
Yeah, I mean, if there's one or two people that want a survey there, there's no reason why not. So, and it's actually good to have some, something that we know is positive, so that we actually say, hey, the positive are coming out positive. So there's a lot of value.
I'll be
in
doing
it.
head and home, right around head on my.
And I'm also Forest Service, how many packets would
You can take as
think
many
would
as you
make
want.
it worthwhile to mail it directly to you
Would-
if we did it on a time that isn't the next week?
Well, I mean, it's, take 5. That's 50 samples.
Okay. And that would be worthwhile, that we
Yeah,
could mail
I mean,
that to you?
yeah.
Okay. Great.
Okay, I guess the only other thing is putting the collection bins at the locations at this point. So you're going back to Hayfork.
Not
Or you're not
until
going
Monday.
Okay.
I work at the watershed center, but,
Right.
well, actually, if I go to the Blitz, then I would be going through there. So I can take responsibility for the watershed one.
Okay, so we have Weaverville, Hayfork, and potentially Northern Trinity Lake area covered. The only places we really won't have bins until Monday would be Mad River and Big Bar.
Well, and the Big Bar is the post office, so it would be, couldn't drop them off until Monday.
It's not open on Saturday. Or Sunday. So I guess, so we can distribute 3 of those, and then we'll try to get the other 2 when I pick them up, samples on Monday anyway. So should we set up any sort of formal sampling, figure out where people want to sample, or people can just sample and we'll?
I would just let people sample freely. I mean, I don't know. I mean, you get, if you amongst yourselves want to decide, want to split areas, that's perfectly, I mean, the only drawback that I see is everybody goes to the same area. That's the only thing, but normally it doesn't happen very often. It's just normally, but you're lost to city, people just go to different areas.
Okay, how do I get?
So I don't know, if you want to circulate, if you want to circulate a map and people want to put like across where they think they're going to sample, so then you're going to get an idea.
You get the
You know.
presentation I had on there. Okay, I appreciate your. Okay, so if we want to pass this around. I think I've heard that, but I'm sure it may work. I got heard that. Okay, does anybody have any last questions?
Yes.
Yes.
Somebody talked about getting certification that you're
That you're
SOD free.
free of something.
Yeah, so that's kind of important because if you look at the quarantine restrictions, even if we're on the east side, I mean, if the guy wants to sell firewood or move wood, you're still bound to, you can't even move it out of the county.
You can move to Humboldt County.
It's,
But I thought that the Board of Forestry had split the county in 2, Pete, wasn't it?
Well, actually, Ryan, what you said was the first I had heard of that.
Yeah.
So I would like to get that confirmed.
Yeah.
Yeah, I'm not,
So what
the
I-
only thing I've heard was the Board of Forestry had, and
this
not
is-
quarantine the entire county.
This is basically what Don
But-
Owen was talking about.
Interesting.
well,
Okay.
Is that with these, can we?
Anyway, because I know that's something that they've been wanting to do, is to
But so,
split the
but,
county out.
Well,
so I can tell you, I just got my exemption to sell firewood, and what they told me was that I'm obligated to follow the quarantine regulations, but CAL FIRE is not enforcing it.
Ah, okay. Well, that makes sense. It really does.
So the firewood I can sell anywhere within a quarantine area and doesn't have to be kiln dried. But if I want to move it out of a quarantine area and sell it in Reading, it has to be kiln dried. So I'm obligated to follow the rules, but the Reading office of CAL FIRE is not going to come check up on me.
Makes sense. Really, in the end, the ultimate arbiter of this is the county ag commissioner.
Yeah.
He's the one who has the
And he's the one who
responsibility.
would give you the
Is there any
free
data from this
designation.
area yet? Has there been any sampling in Trinity County?
No, just the only sampling is right there where.
Right there at the
Yeah.
The only place right
Okay.
here.
So everything that it's generated is going to be new. And that's why, I mean, the data that you generate, if you go to the right habitats, so it makes some sense, and everything, you know, everything comes out negative, this could also have an influence on the decisions that the Board of Forestry, you know,
But
makes
that's
in the
not,
future.
yeah, that's, I mean, when you apply for your exemption, there's nowhere where you can say this is free from anything. The CAL FIRE, and you have to go through the Department of Agriculture and get your mitigation plan or free from the
Well,
Department
if
of
you're
Agriculture.
within the, if you're within the official zone of infestation, that's gonna be the case. But if indeed the county is gonna be split in two,
It is.
like it was originally said, then it would make a difference.
Not what I call CAL FIRE.
Well, but you know,
Might give Don Owen a call.
Yeah.
yeah, but the same thing is,
I think the person to contact is John Owen. Who's the Chris? Who's the guy in?
Lee.
Chris Lee.
Chris Lee, oh, yeah,
Yeah.
absolutely.
Is also somebody good to contact.
Well, because anyway, if you're selling something that's not necessarily a host, you know, like you have any timber operation, or you're selling wood that's not a host, you still have to show that you're not spreading the disease, and that's where the free from is useful. So,
But I don't have any Canoga.
well, then you don't need it.
I need a pencil before we go any further. A pen does not write on it.
I got mine to work on it.
Is there anyone in the
There's a
room
pencil right
who's
there.
planning to go to High Palm area or Pelican Ridge, Southport?
Okay, I'm gonna turn this off.
I think
Are we done?
Yeah, I mean, I guess my impression from Don Owen is that this area, that forest, Cal Fire Forest District is the place where they're actually enforcing
Yeah.
restrictions.
That's what I had heard, that's what I had heard originally
The
when the
entire
Board
county's
of Forestry met.
quarantined, but not a whole lot is being done on this side. And I guess that's,
Yeah.
I mean, the approval
That's
for my
the big-
exemption came with this, you have to follow the rules stuff.
Do you have spatial
But
data for this
Like I said,
line
the best,
or the?
I think the best thing
Can I
to
get
do is
spatial data?
to.
I emailed them.
yeah,
I emailed
file
them and asked
is
them that.
400
That's what he said,
megabytes.
they don't